Alright guys, so today we're going to be talking about the cell. Here is a beautiful picture of a cell. This outside part of the cell right here, this is what's called the cell membrane. This is like the skin of the cell. Now the skin of the cell is what's called selectively permeable. So there's some stuff that wants to go in here but can't because the cell doesn't allow it to go through. Other things go in, out just fine. So one of the things that the skin of the cell uses to do this is called a channel. And so right here, things can go in and out just fine. You also have carrier proteins that will help move stuff in and out. You also have receptors. And so what will happen is you'll have some sort of messenger or whatever that can attach to this and tell the inside of the cell what's going on. The cell can also have cell proteins, and so it will tell the other cells, hey, this is what's going on. This might be in the form of a glycoprotein or glycolipid or some other form. Now, sometimes the cell membrane will also have like enzymes right here. So it might be this is an enzyme that speeds up some chemical reaction. It can be on the inside of the cell, it can be on the outside of the cell. And as I said, this cell membrane is selectively permeable. We're going to allow some things to move in and out. Things that have to use a channel or a carrier protein are things that are charged. That you can just go in and out without a channel or carrier protein are things that are not charged. Things that are lipid soluble like fats, oxygen, carbon dioxide. So another process of the skin of the cell that cell membrane is important in is something called endocytosis. You see how the cell membrane kind of dents in. And so what could be happening here is endocytosis. I'm taking stuff into the cell. Or it could be exocytosis, where I got stuff inside the cell that I'm trying to get on out of here. This thing's called a flagella right here. And so you can see this is a really long thing, really long tail. Pretty much the only cell in the body that's going to have a flagella is going to be sperm. This is what makes women pregnant, is this thing will actually kind of whip back and forth and move the cell up the female reproductive tract in search of an egg that will result in a pregnancy. Here are the cilia. Most of the cells in the body don't have these, but the respiratory system, they'll have them. What they do is they help get junk out of the lungs so we don't end up getting pneumonia. What will happen when you smoke is they actually have chemicals in the smoke to shut down the cilia. Why? Because if you're smoking hey, how long have you been smoking for? About 20 years. That's not going to be very popular, right? Instead, we want a cigarette that you can what a great cigarette without the coughing and everything else. Well, in order to do that, you're going to shut down those cilia. Then what happens is when you wake up in the morning, it's been about 8 hours since you last smoked. And so all these cilia start waking up and start bringing that soot out of the lungs. And so you start coughing. And let's say that you end up in the hospital with pneumonia and happen to be a smoker. What will happen is they'll give you a Nicoderm patch and tell you that you're not allowed to smoke. And a lot of people are like, oh, that's just stupid. I'm just going to smoke the second I get out of this hospital. Why do I got to have a Nicoderm patch? This is stupid. Well, it's not as stupid as what you think. The fact that you're wearing that Nicoderm patch and you're not smoking, these cilia are waking up, but they're bringing the germs out of your lungs. And so that is going to help you get over the pneumonia. The fact that you're not smoking, even though you're going right back to it the second you leave that hospital, makes you more likely to be able to leave the hospital and something other than the casket. Next thing we got is the cytosol. You can see some of this inside the cell doesn't have anything in it. And so that is called cytosol. All this empty space is going to be the cytosol. Now, the little things you see in it, from the little dots to these folds to this great big circle right here, all these structures that we can see that are not just blank space, that is going to be your organelles. When we add the blank space, the cytosol, to the little organelles, everything from this membrane to this membrane right over here is actually going to be called the cytoplasm. This is called the nucleus. So the nucleus is the largest organelle in the cell. And so the outside of the nucleus, you got the nuclear membrane. This is going to be just like the cell membrane. Just like the cell membrane controls what goes in and out of the cell, the nuclear membrane controls what goes in and out of the nucleus. just like the cell membrane had channels, the nuclear membrane is going to have pores that are going to be very important for regulating what goes in and out of that nucleus. This is the nucleoplasm. Basically, it's the cytoplasm, but instead of it being the cytoplasm of the cell, it's the cytoplasm of the nucleus. We call it nucleoplasm. And so this is made up of chromatin, that's your DNA plus your protein. And the center of this is the Nucleolus. This is what reads our DNA and actually makes mRNA. So the Nucleolus has made mRNA, and this is going to be instructions to the rest of the cell telling it what to do. Now, what reads these instructions? So you notice this little red dot, that's going to be a ribosome. And so the ribosomes are going to read that mRNA and are going to make a particular protein. And so the same ribosome can read this mRNA multiple times once it hits the cytosol. Multiple ribosomes can read the same mRNA at the same time. Every ribosome that reads that mRNA is going to make the exact same protein. Now sometimes when the Nucleolus reads a gene, it doesn't make an mRNA that goes to the cytosol, that liquid part of the cell. These are going to be your free ribosomes. Sometimes the ribosomes are bound to another organelle, so those are bound ribosomes. You notice it's on this organelle. So this organelle is called the rough endoplasmic reticulum. This thing is going to make proteins that are then used for export. Proteins that leave the cell and go into the space around the cell. This is your smooth endoplasmic reticulum. Notice all these little tubules right here. But unlike these tubules right here, they don't have these little dots on there. They're not as rough in their appearance. You see it's nice and smooth, right? This is smooth endoplasmic reticulum. This one's the rough endoplasmic reticulum. So the smooth endoplasmic reticulum, it cannot make proteins. Why? It does not have ribosomes on it. So what does this thing do if it cannot make proteins? Well, it does several things. It detoxifies drugs. It stores calcium in terms of our muscles, so when we go to lift something heavy, this endoplasmic reticulum in our muscles just let the calcium leak out into the rest of the muscle cell, which is a signal for the muscle to contract. It makes fats and steroids, so this is what makes some of our hormones. This thing that looks like pancakes is called the Golgi Apparatus. I liken this to the Amazon.com of the cell. So what's happening? Well, does Amazon.com really make anything? No, it doesn't. So then why do people use Amazon.com? Because of shipping and receiving. We're constantly pulling things out of the cell and putting things into the cell with this Golgi apparatus. One of the nice features on Amazon is it lets you gift wrap Christmas presents. Or gift wrap birthday presents. My mom, she lives in Virginia or Texas. I don't have to have this thing come to my house, gift wrap it, and then pay shipping again to mail it to my mother. No, I can click the box, hey, I want you to gift wrap my gift and send it to my mother directly. Well, this Golgi apparatus is getting proteins from the endoplasmic reticulum, and it might gift wrap them with a carbohydrate. And so just putting those little tags on there to say where exactly belongs to the cell membrane, where exactly belongs to the space outside the cell. You got these little blebs here. Sometimes those blebs actually just become part of the cell membrane, and so it's constantly renewing that cell membrane. The next couple of organelles are all unfortunately going to look the same. This is a peroxisome, this is a lysosome. You know, really there's no structural difference here. What matters is its function. So peroxisomes make hydrogen peroxide. And so what do we use hydrogen peroxide for? Well, we use it to kill off germs in our skin. Same thing in the body. We actually use the peroxisomes to actually make bleach, believe it or not, inside of our cells. So very effective at killing germs. This also has a role in starting the breakdown of fats within our body. This is our lysosome. Once again, this is a functional, not a structural difference. So think of Lysol, we use that to kill germs. And so your lysosomes have strong digestive enzymes that they use to break down things. We can use it to break down germs, we can use it to break down food, we can use it to break down waste. Sooner or later, guess what? This endoplasmic reticulum, the rough endoplasmic reticulum, parts of the smooth endoplasmic reticulum, part of the Golgi apparatus, mitochondria, whatever, are going to go bad, and we need to be able to break those things down and get rid of them. Well, this is the guy for the job, the lysosomes. It's going to have those digestive enzymes for it. Last but not least, you're going to have vesicles. And so these are cellular bags. When we want to store something, we can put it in this little bag here, and now we can move it around as we see fit. The next organ I want to point out are going to be the cytoskeleton. So if you look over here, this is a microtubule. This is a microfilament. These little tubules, filaments, and stuff like that, right? Here's your microtubule, here's your microfilament, gives the cell its shape, helps the cell keep its shape. This cell over here, the shape isn't too complex, right? It's just like a glob. Just like a glob right here. But some cells are extremely complicated. For example, your neurons have like this really branched appearance and everything else. Well, how does the neuron keep that branched appearance? Well, it better have a skeleton that helps it do it. The center of our cytoskeleton, these little circle things, these are going to be your centriole. So each of these is a centriole, but as a group, there's the centrosome. And this is going to be the backbone of our cytoskeleton. Centriole, as a group, centrosome. Not only do they have a backbone of the cytoskeleton, it has a role in cellular reproduction. Here's cellular reproduction, we call that mitosis. And so right here, here's your centriole, right? Your centrosome. It splits, moves to opposite sides, you make these spindles. The chromosomes are going to line up here in the middle in metaphase. Those spindles are going to pull them apart as it goes back to the centrosomes. That's anaphase. And now that the spindles are done with their job, the nuclear membrane is going to reform in telophase. But notice what had a very key role in this whole process. It was the centrosome and the cytoskeleton. We've been talking about endocytosis, bringing things in, exocytosis, kicking things out, the manufacturing center of the cell, which was the endoplasmic reticulum. We're making hormones. Where are we getting the energy for all this stuff at? Well, what's doing this is going to be this organelle right here. This is the mitochondria. And so the mitochondria is the power plant of the cell. And so what this does is it takes our food, fat, proteins, and carbs, and sucks energy out of it. And then it stores it in a way that our body can use it. And so that form is going to be ATP. Now, without the mitochondria, you can get some energy out of your food. It's just not very fuel efficient. So one glucose molecule with the mitochondria produces 34 ATP. One glucose molecule without the mitochondria only produces 2 ATP. So we're getting 17 times more energy out of our food using that mitochondria. Secondly, the mitochondria has a role in cell suicide. So if you're a cancer cell, you don't want these things. Why? Because it might make you die. And so a lot of cancer cells will get rid of the mitochondria. But without the mitochondria, off the bat, you're having to use 17 times more glucose because you're not producing a whole lot of ATP from glucose. Then on top of that, what happens? Well, without a mitochondria, you cannot break down fat, you cannot break down protein. And so you're very, very limited in terms of what you can do. So you're going to have to use at least 19 times more sugar than a normal body cell. We can use this to our advantage in terms of diagnosing cancers. Now, a couple of things I want to point out here before I close today. One of the things is, as I said, you have one nucleus, looks like you had one endoplasmic reticulum, one Golgi apparatus, and so on. But you can actually have more than just a set number of organelles. Typically, you're just going to have one nucleus, and the endoplasmic reticulum and Golgi apparatus will get bigger and smaller as you need it. But the mitochondria, you can actually have more or less of these depending on the use. So, for example, if you're a muscle cell and you're just constantly running marathons all the time, you're going to have more mitochondria and bigger ones. Look at this fictitious cell. We've got 1, 2, 3, 4, 5, 6, 7 different mitochondria. Same idea with peroxisomes, lysosomes, and vesicles. You can actually have more than one. One more thing to point out is your vesicles can actually fuse with the lysosomes. And So we use this to break down bacteria.